Yo, and welcome to Shooting the Soil. This is my end of August tour of the garden. We're going to do things a little bit differently this time around. We're going to show you one bed and what's growing there, and then we're going to look at it under the microscope. And then we'll move on to the next one and compare what everything looks like. So today, rocking my Straight Outta Compost t-shirt. Shout out to San Diego Seed Company for the shirt and the extra packet of jalapeno seeds that you sent me. Appreciate it. And with no further ado, let's dive in. Hey, so right before we get to the tour, I just wanted to share a big announcement with you. I just signed up for Matt Power's Regenerative Soil course. And so basically I'm going to be able to give you a much more educated perspective on what we see on the microscope, and I'm going to be able to take much more educated actions out in the garden, so the best is yet to come. And sorry I haven't got too many videos out lately, so now you understand why. Been pretty busy with learning all this information and trying to wrap my head around everything, but it's pretty fascinating. So yeah, let's get to the tour. Alright, so here's my lower bed with my cucumbers and my peppers. We're going to check out how this soil is doing first. And as you can see, we got some little pepper plants in here. Basically, with the drought and everything that we had in New England, I don't think I watered enough. And they really just didn't grow that much, but they're starting to bounce back. This poor cucumber's having a rough time, but I think they're gonna bounce back. We'll get a couple more cucumbers out of these before the end of the season, I do think. Yeah, we actually had some blossom rot, so a lot of our cucumbers did not grow through August here. But we did get a couple through July and spring that were extra tasty. Alright, so that's what's going on in the lower bed. Let's see what it looks like under the microscope. Okay, so here we have the lower bed, and we're seeing a whole lot of bacteria. Mostly coccyx, the round-shaped ones, but a good amount of bacillus, the more elongated ones. And we got this super fast moving one here, which is bacillus shaped, but I don't know if they move that fast. It could also be a spirilla or a vibrosis, hopefully a spirilla. I found this interesting amoeba here. Got a Mercedes symbol on them. I believe that's an amoeba anyway. Pretty cool looking. But ultimately, despite the nice aggregates and a lot of bacteria, the main problem I'm seeing is not much fungi. Here's two little fungal spores, and below it, a very clear strand with sort of visible septas, but not necessarily a beneficial, but it could be. And here's another clear fungi strand, but this one's looking a little bit more equal and uniform and nice clear septas. And here is the best fungal hyphae that I found on the entire slide. We really didn't see that many, but anyway, we see the nice septa here, decent brown color, and it goes all the way up here into this other aggregate. But then, wait, right above that, we see some strands coming out that don't look all that beneficial. Not quite sure what they are, but I do think I really need to work on this soil, and hopefully we can get it better next year. And we found one nematode on the slide. Hopefully this isn't a fungal feeder. That would kind of explain why I got no fungi. But I don't think he is. Let me know in the comments if you have any idea what this nematode might be, or what type. And we'll take a quick zoom in on the mouth here, because that's how they're identified. And I'm leaning towards bacteria feeder. And now, back out to the garden. And now on to our cherry tomato raised bed. And we got a lot of tomatoes, and one has made it to the top. Was hoping they would be all up top by this time.
But they're still growing. We still got some time left to see if we could get a canopy here of cherry tomatoes. In the meantime, I think I gotta harvest all these tomatoes to promote some more growth. And let's see what this soil looks like under the microscope.
And here we are in at 400x, and right away we got a fugly looking strand here that it's not very uniform, very clear, no septas, so not sure if that would be beneficial. Most signs I know would say no, but it could be. And here is a big old hyphae. All signs point to this one being beneficial. But like the other sample, I didn't find very many. Probably even less fungal hyphae than the last soil, I would say. So I still got a lot of work to do on my fungal to bacterial ratio. And speaking of the bacteria, I was actually seeing a lot less bacteria in this soil also. And here we have a cool colored cyst, or possibly a small amoeba in the center of the screen. But I really saw no other ciliates or nematodes or any other microorganisms in this sample. I think this raised bed design, with the chipmunks digging little holes through it and trying to make a home, led it to be a little too oxidized. And now back out to the garden, to the bed that, to my surprise, performed the best this year.
And we cloned and planted some strawberries in these little pots and stuck them down so I don't accidentally drag the hose over my artichokes and wildflowers over here. These two artichokes are looking the best, despite this being the lowest bed and a lot of clay, only a couple inches down, but they don't seem to mind it. They actually seem to like it. And it's part of the reason why I planted the artichokes here, because they have a deep taproot to break that up. And we got a couple of wildflowers sprouting here and there. They're doing all right. And so let's check out what we got over here. Our basil really exploded. I've just kind of let it go to flower because, as you can see, all the insects and bees really like it. There's a bumblebee, and I think I saw a honeybee around in there somewhere, but alright. And also in this bed, our Swiss chard is looking quite nice. See all those really vibrant colors and everything down in there. Been making a lot of juices with this, and it is just blowing up. And over here we got our second run of peas. We harvested all of the peas and made a really nice pea soup. We'll give you a look at that here. And now we're going to take a sample from right over here next to this Swiss chard and see what's going on under the microscope as far as the biology goes here.
Alright, so right off the bat here we have a really big nematode. We're in at 400x, the same as the last nematode we looked at in the other sample. And this time we'll freeze frame to zoom in on the mouth parts here. And I think this is an omnivore. Let me know what you think in the comments. With that tri-tip nose, he's got to be either a really big bacterial feeder or an omnivore. So we saw a little bit more fungi than the other two samples in this particular bed. Here's a very nice uniform one, nice and long as well, with nice septies, but not super dark, so he's probably just a young one. And then, of course, right over here to the left, we have a not so beneficial looking strand, but gotta show the good and the bad, so we all can learn from it. And taking one more look at that nice uniform strand that we are just looking at, and I wanted just to point out in this shot how much more bacteria than there is in the last one, and I think more than even the first one. And here's another look at all the bacteria. Didn't see any of those big fast-moving ones in this bed though. And to the right there we have a kind of beat up fungal strand, but a great color to it. And we went back and found that same big old nematode that we were looking at before, so we can capture him in the dark field. We are out at 100x right now, and I really like the dark field for getting an idea of your soil aggregation, since everything that's aggregated glows orange and brown, and everything that isn't glows clear. But my favorite part is definitely how all the bacteria twinkle like stars as they bumble about, and make it look like an image from a space telescope instead of my microscope. And here is a look at the 3 soils in 1x, going from left to right in the same order we showed them. And all 3 of these beds were made from the same soil that I purchased locally. Basically a mix of local topsoil, mulch, and compost. Knowing what I know today, I would have done more sourcing on the compost. But at least it's local soil, I supported a local business, and you guys get to see me build it into a really good soil. And I should mention, all these beds were watered the same just the day before taking the sample. Zooming in here on the lower bed, we see a pretty nice brown color, had some pretty nice structure, but still a little silty. And moving on to the raised cherry tomato bed, as you can notice, it's much drier than the previous one. A lot of clay showing, which kind of surprises me since they're all the same soil, how this one got so much clay in it. Not very good soil structure, and appears to me to be getting very oxidized, either with my poor bed design and air getting in through the bottom, probably a combination of that and the chipmunks that dug a bunch of holes in there. And Swiss chard bed has the best color, best soil structure, and the most water retention. Alright, so now what did we learn from looking at all these soils under the microscope? I think one of the biggest things we see under the scope is how we can improve, and from these samples we definitely need more fungi. We saw a big decline in fungi throughout the drought, so we're going to have to work on our watering through times like that for sure. And also, I think I'm going to experiment with a lot more mycorrhizal fungi inoculants and stuff like that. And since the cherry tomato bed was the worst in pretty much all categories, needed watering the most as well, we definitely got to redesign that bed somehow. And what happened to all the protozoa? We didn't see any protozoa in any of these soils or ciliates. And we definitely saw a bunch in the compost teas and extracts that we were applying. So I'm going to learn a lot more about diagnosing all of these problems come this spring once I finish up my course. So with the regenerative soil way, we're going to make this soil better and better every single year. And right before we wrap things up, here is the area of New England wildflowers I had planted and mentioned in a previous video. A couple of the flower species did really well. Gonna seed again next year and hopefully get a little bit more diversity. Well, that wraps up our garden tours for the season. Next season, we're gonna be doing regenerative soil home gardening. But don't worry, we got some microscopy videos planned for the winter. So hit those buttons and help grow the channel if you would be so kind. And I'll leave you with a shot of Lilu being silly after a swim.
Is that a good swim?
Thank you for watching, and catch you on the next one.